If you really want to understand any disease, I'm talking cancer, heart failure, you name it, you can't start with the organ you can see. Nope, you have to go smaller. Way, way smaller. You've got to travel to ground zero, the single individual cell. It's right here in this incredible microscopic universe that the story of our health and our sickness truly begins. So let's dive in. The word pathology literally means the study of suffering, and the absolute foundation of all modern medicine is this one simple truth. Every single disease is just a cell that's in trouble. But what does that really mean? It means a cell is stressed out, it's gotten injured, or it started making mistakes. And that process, well, that's the key to everything. Okay, get ready for a fact that might be a little humbling. Inside every single one of your trillions of cells, you've got a genetic blueprint with about 20,000 genes that actually code for proteins, the building blocks of life. But here's the kicker. A simple little worm with less than 1,000 cells also has about 20,000 of those genes. So if it's not the number of recipes in the cookbook, what on earth makes a human so vastly different from a worm? We are absolutely going to solve that mystery, and our journey is going to take us deep inside the cell. We're going to explore the genome's hidden dark matter, we'll look beyond the DNA sequence itself, meet the cell's amazing housekeeping crew, and then we'll see what happens when that blueprint goes wrong, and how tiny, microscopic errors can lead to major, devastating diseases. So let's start with that fundamental idea. To study suffering, we have to start with the cell. All right, our first stop is the cell's command center, the nucleus. This is where the master blueprint for everything is kept. But what we're about to find in here, well, it's not what you'd expect. Turns out the biggest secrets are hiding in plain sight. Get ready for a number that should honestly blow your mind. Of the 3.2 billion letters in your entire genetic code, only a tiny, tiny fraction, just 1.5%, is actually what we think of as genes. The actual recipes for building all the machinery of life. Just 1.5%. So you have to ask, what is the other 98.5% doing? For decades, scientists just shrugged and called it junk DNA. Can you believe that? Junk. But as it turns out, that junk isn't junk at all. It is the genomic dark matter, and it's where all the real magic happens. And this is the incredible answer to our worm question. It's not about having more ingredients. It's about having a master architect who knows exactly when, where, and how to use every single one. This so-called junk DNA is the incredibly complex architectural plan that directs the construction of a human instead of a worm. So what exactly is hiding in this genomic dark matter? Well, it's the entire control panel of the genome. We're talking about things like promoter and enhancer regions that act like on-off switches for genes. You've got these master controllers called non-coding RNAs, and even jumping genes that can literally move around the genome. This is where all the regulation is, and regulation, that's what creates complexity. But wait, the story doesn't stop there. Knowing the DNA sequence is just step one. The real question is, how does the cell decide which parts of that massive blueprint to actually read at any given time? This, my friends, is the world of epigenetics, and this is where it gets really, really cool. I want you to think of it like this. Your DNA is the script for a play. It has all the lines. But epigenetics, that's the director's notes all over the script. It doesn't change the words, but it completely changes the performance, telling one actor to shout, another to whisper, and another to stay silent backstage. And incredibly, these notes can be passed on when the cell divides. Okay, so how do you fit 2 whole meters of DNA into a space you can't even see with the naked eye? It has to be packaged with unbelievable efficiency. The cell wraps it tightly around these little protein spools called histones. And this whole DNA protein bundle is called chromatin. Now here's the key. This packaging isn't static. It can be loosened or tightened on command. This dynamic packaging creates 2 basic states. When the chromatin is all loose and spread out, it's called euchromatin. Think of it as an open book, ready for the cell to read. But when it's packed down super tight and dense, it's called heterochromatin. That's a closed book, silencing every gene that's locked inside. So how does the cell know when to open the book and when to slam it shut? Tiny chemical tags. They act like little sticky notes that get slapped all over the DNA and the histone proteins, telling the cellular machinery, hey, read this part, or nope, ignore this section. These marks are the physical switches of epigenetics. Now, this is all fascinating biology, for sure, but you might be sitting there thinking, okay, so what? Why should I care about this? Well, this is where things get incredibly exciting, especially when you think about the future of medicine. And here it is. This is the amazing part. A mutation, a typo in the DNA sequence itself, that's permanent. It's written in ink. But these epigenetic marks, they're like writing in pencil. They can be erased. And that reversibility is a massive, massive deal for medicine. For instance, some cancers work by epigenetically silencing the very genes that are supposed to stop tumors from growing. Well, guess what? We now have drugs that can go in and literally erase those silent marks, reopening the gene and letting it get back to work fighting the cancer. How cool is that? All right, so we've spent some time in the cell's front office, looking at the master plans. Now let's head out to the factory floor, the cytoplasm. We're about to meet the crew that does all the heavy lifting, and just as important, takes out the trash. A cell is a nonstop construction zone, always building new things and breaking down old ones. So to keep from being totally buried in its own garbage, it has two main disposal systems. First, you have the lysosome. Think of this as the heavy-duty recycling center. It's basically a bag of powerful enzymes that can digest just about anything. Then you have the proteasome. This is more like a precision paper shredder, specifically designed to find and destroy individual proteins that are faulty or just past their prime. Now, when a cell gets really stressed out, like, say, during starvation, it can do something truly wild to survive. It literally starts eating parts of itself. This process is called autophagy, or self-eating. The cell wraps up non-essential components in a membrane, fuses that package with the lysosome, and then digests the contents for raw materials and energy. It's the ultimate survival mechanism. And of course, we have to talk about the mitochondrion, the famous power plant of the cell. But there's a fascinating twist to its story. It doesn't just generate energy. The mitochondrion is also a sentinel. It's constantly monitoring the health of the cell, and if things get too bad, it holds the keys to the self-destruct sequence, triggering a clean, controlled demolition called apoptosis. It is a true double-edged sword. So we've seen the blueprint, the regulators, and the machinery. Now it's time to connect the dots and see what happens when this whole beautiful system starts to break down. This is where the microscopic world of the cell crashes into the very real and often devastating world of human disease. Just imagine that factory again. One of the machines starts spitting out faulty parts. At first, the quality control crew, our proteasomes, can probably keep up. But if the problem gets worse and worse, pretty soon the factory floor is just clogged with junk, and the entire operation grinds to a halt. That is exactly what can happen inside a cell when misfolded proteins start to pile up. And believe me, this isn't just a theory. Let's look at two devastating real-world examples. In some forms of retinitis pigmentosa, a single protein in your eye, called rhodopsin, starts to misfold. This accumulation leads to the death of your photoreceptor cells, and eventually, to blindness. In Creutzfeldt-Jakob disease, misfolded prion proteins trigger a catastrophic chain reaction that just destroys brain cells. It's a terrifying reminder of how one tiny molecular error can change a life forever. So we end with this question. The cell is an absolute masterpiece of resilience. It adapts, it recycles, it has multiple ways to die cleanly for the good of the organism, but we all know this intricate dance doesn't last forever. If our cells are so brilliantly equipped to survive, what is it that ultimately ensures their decline and leads to aging? Well, that's a story for another day.